Well, I guess thanks very much. Yeah, my name's Renan. Today I'm just going to give you a little bit of a talk, about kind of the work I used to do for the Ministry of Health. But before here, last year, I used to work for NSW Health Pathology, where we delivered this pathogen genomic service. Um, and then I'm going to be, kind of trying to first focus a little bit on what the service was about and what the government needed it, and then going into how we actually implemented the service by by implementing Nextflow and making things actually deliverable. So how do I get that bar? Oh, it's gone. Okay. Oh, you can also see this. All right. So start by just giving, I guess, a brief overview of why the government is interested in this. So there are pretty much just these three pillars, three areas where the government is interested in. One being for public health response. So we want to be able to detect outbreaks early for stuff that is dangerous to people like COVID, TB, tuberculosis, salmonella, things like that. And we also want to be able to trace the source. So we can stop outbreaks before they actually happen. And then once we get that data in and that evidence available to epidemiologists in the ministry, then they'll go on to become policy and regulation. So like an example of this, for instance, is for countries that don't have certain bacteria haven't been eradicated yet. So for example, Brazil, I'm from Brazil. So from Brazil, we still don't have eradication of TB, for instance, which is a bug that the government is particularly worried about. So this service helps the government kind of know when these bacteria arrive in Australia, where they're coming from. And then in countries like Brazil, when you start seeing lots of people having this, the government just says one of the policy will be, all right, before you come here, you actually need to take a test to make sure you don't have it. So it helps guide these kinds of policy. And then another branch that is also really important for hospitals and then for GPs in particular is just the antimicrobial resistance branch, which is a branch where once you get a sample from a patient in the hospital, the bacteria they have might be resistant to certain antibiotics. And we want to know, before giving them a random antibiotics, we want to know exactly what the bacteria is resistant to so that we don't give them something that doesn't work. And then for the talk, I think we're going to be focusing, well, the policy part was actually out of my area of expertise because that's up at the ministry. Epidemiologists would do that. And then my job was just to deliver the data for public health response and AMR, which is antibiotic resistance. So then we're going to be talking about a little bit about these two areas. Because the epidemiology, really what we want to know is where did the sample came from, where did the isolate came from, and where is it going? And we can tell that by analyzing the DNA of that sample. And I'm going to briefly go through the steps of how you do that bioinformatically. And you also have the other branch, which is antibiotic resistance, in which case, we're just going to try to, once we have the DNA of that sample, can also tell just by matching that against a database of known genes that are linked to resistance, we can tell if the bacteria is going to be resistant to a particular drug. And so then we're just going to start talking a little bit about the, I guess, surveillance kind of part of the talk, which is just the epidemiological branch. And then usually you have a workflow like this, and then the pipeline is going to start with you having your DNA reads here as the raw reads representing there. And then after this, my job or our job was to just go through put these samples through the QC, just make sure that the lab gave us the right samples, and then there isn't any issues with the reagents they use. Just make sure that the samples are intact and then good quality. And after that, then we start the bioinformatic analysis to try to be able to tell that's going to first align this DNA to a reference so that we can get SNP colonies, some variants there. And then once we have these variants, we're going to be able to produce this phylogenetic tree, which is just another term for cladogram for the statisticians. We just have this cladogram that's going to tell which samples are more closely related to each other. And that's going to help us infer where these things came from. So a little bit more about this section here from the green onwards, so that you get a better idea of how that works. So we might have 4 DNA samples in there. And what we do is just want to align those sequences in order to, and then try to find the differences there between them. So for this example, like say sample A and sample B are more similar to each other than sample C and sample D, as you can see by those columns that vary on those samples. And then that's going to lead to that cladogram, where A and B are more closely related to each other, and C and D more closely related to each other. So then you can imagine that we might have this 1 week, and then if for the following week we were to get 10 samples, and all 10 start grouping with the clay green there, then that means that the bacteria is spreading, and that's particular type. And because we know where patients A and B came from, and we know that they're the 0.0, then we can start kind of delineating a map of where that's going and where it started. And that's going to help then the ministry try to implement policies and breakpoints to try to halt that infection, the spread of the infection. And then another way that another interesting thing that we can do as well is by is being able to tell whether a particular sample came from within Australia or maybe from another country. So a similar approach, you're probably familiar with this, is just those ancestry tests that we have. 23andMe is a company that is got famous for doing this. So the way this works is that they have a database of samples whose heritage is known to them. And then they just got to match the DNA. If you send the DNA to them, they're just going to try to match. They're going to do this, get this NIPS. Once they get this NIP profile, which is just getting all of the positions and the bases that vary in which one of the columns, they're going to try to match to the database. So in this case, the DNA sample matches perfectly with the European. So let's say I have 4 bases. And let's say that if you're in your example, you have actually 10 bases. So 4 of them match perfectly with European. If the other 6 were to match, I don't know, with African, then you say 60% African heritage, 40% European. So that's how they do that. And it turns out that if you can do that to humans, you can do to any living thing. As long as you have DNA and RNA, we should be able to tell. And this is actually how we do it for an actual pathogen. So this is just, I can actually show the data that we have because that's confidential, I guess. I also don't have access to it anymore. But this is one paper where people actually did this, the exact same thing. So this is an example of a pathogen, Salmonella Mississippi, which has 3 specific clades. We got AYY here in purple, which is endemic to Australia, as shown by the purple ring there at the top. We got the red clade here, which is AI, and then it's endemic to the US here in light gray, and the one at the bottom is endemic to the United Kingdom. So then after they do this analysis and they do that clustering, so if you were to look at one particular example, you can see that this sample green here was actually sampled in the United Kingdom, but it clusters with the United States. So then from here, we'll just make a report, send that to the ministry. The AP will check the file from this patient, and then they interview the patient right in the hospital. And it's like, oh, you've been to the World Cup, right? Yeah, I did. And then be able to tell that came from the US. That makes sense. And then if you were to start seeing more cases of that, then the ministry would be interested in knowing whether that came from that patient or if all of them just came from the US, from the World Cup. And then it's going to dictate what the reaction should be or the response should be. And this is an actual example that we work. This was before my time, actually, in 2016. But as I was there, we kept going this service for Legionella, which is this bacteria that grows in water reservoirs. So whenever you have water reservoirs there, there's Legionella. And then the government has some water towers, and they're owned by the government. And then we know that from time to time, we get Legionella infections on them. And then people show up in the hospital with those infections. So this is just an example of a paper. So then our job there is to try to tell where they got it from. Usually it's from one of the towers. And then we have a database for the towers that have been whose samples have been previously sequenced. So then all of these that are colored are just towers or clinical samples that have been extracted from CBD or from these 2 suburbs that is showing there. And then our job pretty much is to try to see if the clinical samples group or cluster with any one of the towers that we have in. And then in this case, it's tough to say, it's tough to tell. But here, the clinical sample are kind of labeled with a C. The environmental ones are labeled with an E. So then you see like these 2 clinical samples are grouping with these environmental samples, towers 6 and 5. So then the government, rather than just sending it to all of these towers, just send people to these specific towers where you know the Legionella infection is, and then just try to clear that out. And this is a problem that is like our public health interest, because you can get this by just breathing near the tower. It's just like Legionella just goes in the water droplets. Sydney is really hot. But then if you just breathe that in, you get that. And then it's public health hazard, so it needs to be taken care of by the government. So that's one of the services we had specifically for Legionella. And then the second one, the second part of the talk, I guess, another work that we did is antibiotic resistance, which works the same way. We kind of get the DNA samples from those bugs, we do the quality assessment, and then we have these 2 other steps here. It's called the assembly. So it's one well, we can't really sequence the entire DNA of the actual bug. It's too large. So what the technology at the moment does is that it breaks it out like a puzzle. And then we get those puzzle pieces. And then these packages that we use can actually match those puzzles together and make the chromosome, the entire chromosome in one piece back to us. Or not in one piece, but for the sake of this talk, I'm going to say it's one piece. And from that, we can then do the kind of similar thing that we do for the surveillance part, which is to just try to match DNA sequences to a database of resistance genes that have been already published by the academics. So, for example, here on the right, I have genes for that confer resistance to these different drugs in here. And then what we do is that once we get the assembled genome of one of the samples, we try to ask, or does this genome has the sequence for BLAST-CTX, for example, which confers resistance to beta-lactam? And then if that's true, then it can give that patient this antibiotic. And the reason that that's really good that we do, well, before back in the day, we used to do this by phenotyping the strain. So like in the lab, they'll just get it from the patient and then they go to the lab and expose that bug to the antibiotic. So if the bug keeps growing, it's resistant. If not, then you gotta try another antibiotic. But that takes a little bit of time. And for specific bacteria like TB, you can't do that because it takes 6 months to grow bacteria TB in the lab. But then we can tell this in the same day, so same day delivery. So that speeds up that process for the hospital as well and for the GPs that requested it. And usually, if you have a close match, either higher than 90% identity just means a full match or exact match. Then we're just going to say that bug is resistance to this bug. So this particular example I gave here, isolate one, would be a particular worrisome one because it's resistance to almost everything except vancomycin, right? And then if you were resistant to, like, let's say we only had these 5 antibiotics and it was also resistant to vancomycin, then you're on your own kind of thing, right? Either your body gets rid of it or you die eventually from it because it can't really help you. So that's why government is really interested in this, so that we can give you the right type of antibiotics so that bacteria don't evolve resistance to all of them at the same time. So then we'll actually give them a heat map that looks similar to this, which is, so here on the left to the cladogram, we just have different samples of Klebsiella, which is also a bug that gives you pneumonia, that we also track. And here at the top, I can't read it maybe, but here at the top, we got different antibiotics. Here are the genes that give resistance to these antibiotics, and then we try to match. So if you were to go to one of the antibiotics here, so this black part that shows the phenotype of the sample, meaning they tested this in the lab and know for a fact that it's resistance to these. And this is what the genomic prediction gives. So if we were to look at an example, here's penicillin, and all the bugs are resistant to that because it took so long. And then when we look at the penicillin box, then all of them have at least one of these 3 genes. And when they don't, right, it shows that the phenotype is there, but the genes aren't there. And that means one of the 2 things. Either they have a gene that is not present in the literature, or more often, this step here, it means perhaps that they have the gene, but it's at 89% cutoff. And then the fact didn't get it because we have a specific threshold. But then for our pipeline, because we know this ahead of time, then we have the pipeline flag it. If it's like, oh, it's higher than 50% or a particular percentage, it's not going to show up as a completely full cell in there, but it's going to trigger the bioinformatician to just go in there and check to see if that's a real result or not. And oftentimes, yeah, it's not. And then we're just going to have to go there and then input that manually and then let them know that the assembly wasn't great, or just try to use a different assembler on this part that's going to maybe assemble the puzzle a little bit better and perhaps give us the full gene. And then here for the service, we actually had so every week we had about 200 samples that I had to process. 100 of them were COVID alone, and the other 100 was a mix of different bacteria in there. And all of these studies I showed before, right? They're usually PhD students, masters. So it takes 3 years to just process those 20 samples or something. But then if you're processing 200 every week, then you need to have something that automates this for you, as you actually don't have the time to actually go through all these specifics. And then that's when Nextflow comes in and workflow managers. So the Nextflow is one type of the workflow managers that manages all that pipeline for you. So you can set it up once, and once you have it well set up and well tailored to the type of data that you have, then you can run it. You can just run it every week, and then you just handle everything for you without a lot of intervention. And then the bioinformatician really just need to intervene when there's some questionable results there, which will be flagged by one of the modules in the Nextflow, if you code it properly. And then this is also the seminary. And then we can do this not only for pathogen genomics, but for anything that our professors here, NSW and BABS or academics usually ask for. There's usually a Nextflow workflow already publicly available for that. But if it's not, we can just create one. And then it's great for service that once we create one for one professor, let's say an analysis that is not there, and we have it on our portfolio, then that's there forever. You can just use it for the next professor that wants the same analysis. And then it's important because of these four pillars that I have in there, skill. And then we're going to go through each one and give specifics on how Nextflow handles this and makes the service effective. So the first one is scalability. So why this is good is that Nextflow is able to enables you to use HPC and then does all of this work of running things in parallel for you without you having to do much. So here's just an example of if you were to run this manually with one sample, let's say you have 4 analysis steps, which one takes 4 hours, that'll take 4 hours. But then if you were to run 200 samples, that'll be 800 hours here. So then you don't have that time. But then if you were to just run it with Nextflow, because it can run everything parallel, then that amount to take 200 samples will still take 4 hours because it's running everything at the same time. So Nextflow will just handle this. We have here, when you're in NSW, have access to Katana, which has the HPC cluster. Over there, the government, we had access to we use AWS, paid like $800 a month for the 200 samples every week. And then they kind of charge you for usage, so depending on how much you used. But then you just run, and then with AWS is good because you know that it's going to run for sure. You don't have to wait in the queue. Sometimes here in Katana, if everybody is running analysis, right, there's a physicist trying to model something fancy, you might get in the queue because they're using all the resources. While here you're just trying to, you just get the resource right away because you pay for it. But that's one. Another one is reproducibility that is important. For government, people used to sue us a lot. So what we needed to be able to rerun analysis with the exact versions that we ran before and make sure that the results are reproducible. So one thing that we started doing also was an internal audit when I got there. So every 3 months, supervisor will come and then you just like pick 200 random samples that we analyzed like maybe a year before, 2 years before. Don't tell us it wouldn't tell us what the samples are, and then you just have to give the exact same results. And then we'll do this by just running the exact same pipeline. And then the way we do this is that Nextflow allows us to use container environments. Usually the one we use is Conda, just the environment you create there, and then you can install the exact versions of the files or the packages that you use for that analysis. But then every time you run, if you were to rerun a sample, you can just, and even if you were to want to move this pipeline into another computer that doesn't have any of the packages, you can just install it with one file. The file has the exact same versions, but install it, then should run the exact same thing that we had on the original computer. And then that's great because this then immune to the audits and make everything easy to rerun and reproducible, so that also a trainee can just run it really, really quickly. Another thing is modularity as well. That is makes it really easy to just swap modules around. So, for example, there's specific bugs that may require different kinds of alignments. Let's say try to do that step or try to align DNA sequence to somewhere. For Legionella, you might use EWA, but if you have Salmonella, it might be it might align things better with multi-2. So then you can just rather than having to rewrite something, you just swap that one module and the pipeline works perfectly as well. So then we really, I guess, we don't have a lot of time there, press for time. So then we always, it's good to have it as efficient as possible. Another pillar is reusage. Because all the time we both need to track the outbreaks, surveys, and sometimes there are specific projects that people want to hire the service for. And then it's useful to be able to get the pipeline and run the exact same thing that we actually ran before, or that we run every day. I think the last pillar is just automation, because we can't do it without so we were just 3 bioinformaticians for 200 samples every week. So we wouldn't be able to run this manually. So then Nextflow makes it very easy to automate. once you have a particular pipeline ready there, then all I have to do is just run the command. Nextflow run your pipeline, and the Nextflow is gonna handle everything. So if a job fails here, Nextflow is gonna retry it by itself, or let you know that this job failed and you need to check this. But then you just keep all the job scheduling, job submitting, everything is being handled by itself, which is good because then we can go on to start writing reports and doing other things. We had our own custom scripts for this because we needed modules to do specific things with particular parameters. We also had sometimes databases that were internal, so we couldn't actually share. But here at Test Central, we'll actually be using this repository, which is called NFCore, which is just a repository of several Nextflow workflows that have, for a majority of different analyses, they're common to genomics or proteomics or whatever professors here at the BAPS and units that we usually use. So then here I just highlighted two of them that do the exact same job that our workflows used to do, pathogen surveillance and FunScan. And this is how I guess a picture of those pipelines would look like. So each one of these white nodes is a module that you write yourself. And once you have them, you can just call them in sequence, and then Nextflow is going to handle all the rest. And this is just taking a look at what one of those modules look like. So like if this was BWA and what you look at what that actual coding inside the module looks like, then all you really need to do is to specify that this is BWA. You just can name it whatever you want. Then you set a tag for the sample ID, which they're calling here metadata ID, right? Meta ID, but it's just really the sample ID so that you know. And then here you just specify the actual resources that you need for that job, so that it doesn't, if you're using AWS, right, you don't end up paying for more than you actually use, because that's taxpayer money. So you just want to set up the exact resources that you actually need, and you can get this from experience or from doing a few tests. And then you also can specify the container that has the exact versions of the software you need to run to make sure that everything's going to run as intended. And all you really need for this step that performs the alignment is just give the input, which is a sample name, and then the path to your DNA files and a reference. So you just align it against the reference so they can call the variant later. And then you specify also the output, which is just a sample name, and then an alignment file called BAM, which we're calling BAM here. And this is important just for our audits, just to have all the versions in there and make sure that that analysis ran with the versions you actually intended to run it with. And here's the actual command. So back in the day when I started training, we actually do this manually. So like I'll just be on my laptop and running this sample by sample, or just writing, I guess, a bash script that does this, but still you just need to run it by kind of manually. But here you just set up the command they usually use with all the resources they already specified, and then it's just getting the reference and the actual DNA files, and then it's just going to perform that analysis for you. And what Nextflow does is, like, after it finishes running this module, it looks for these files. So it's going to look for the file with the BAM extension, and for this particular file called version.yml. Those files aren't there, it just tells you they broke, and then you got to fix it. But if it does, then it says that it completed correctly, and then it goes to the next step. And then the next step might look like this, right? After you get that from the variant calling, same thing, specify the resources. But this time you're getting the path from the previous module, which you know for a fact gives you a BAM file. Because then if this doesn't give you a BAM file, then the step failed. So that's great, because then Nextflow just had this another layer to make sure that things ran correctly. And this will give you, I guess, these NIP files in there. Then you have the command that gives you as well. And then that big workflow then in coding would look like this. It's just really simple. You just have a workflow, and here you're just kind of calling, here you're just specifying where your DNA files are, and then specifying where your reference files are. And you know that this module only needs to take the DNA files and the reference. So you just put those in there, and then variant call takes the output of the process you just ran, and then uses the reference to specify that as well. This is how you do it in practice, and then once you built this once, this is great that if Peter were to come, well, there's another professor who wants to do an analysis, but he needs to swap one module. I actually just copy and paste this, and then if there's a module, they use a different variant calling package. Yeah, just swap that one in, and then that's just one line code, because I already have all the workflow in there. So it's really efficient, and it's great for service. It's been yeah, it's been very useful for us. With the we have the first I think we used was the bulk RNA-seq pipeline, which I have been working on Nextflow now. Works really great and it saves a lot of time. All right, and then so that's a summary. So it's pretty much just showing what we're talking about, why the government is interested in these types of analysis. Just asking if if these cases are connected and which drugs these pathogens are resistant to. And we have roughly 200 pathogens that we have to process a week. So then having Nextflow really helps. And we do this by building phylogenies for the surveillance and for the resistant genes. We just do that analysis so that GPs know which antibiotics to administer to patients. Yeah, and then just using workflows like Nextflow is what actually makes this service possible. Otherwise, if this was maybe 20 years ago, yeah, I wouldn't be able to do it with the technology we had back then. So yeah, so that's how we can implement Nextflow into a genomic service that is useful as to the public and to the government. And yeah, thanks very much. And if you have any questions, happy to try to answer them. And then if you have any consultation requests, just started the Bioinformatics service in January and let us know. We'll be there. Thanks very much.
There's a question in the chat.
All right. Question in the chat.
May.
Yeah.
Read out the question for the.
Yeah. So question is, can I ask if SnakeMake is a comparable option? Is it less production ready than Nextflow? Wonder if I should convert my pipelines. My take is they should absolutely convert it. Now SnakeMake is also very useful, but I guess it was created at the time where we didn't have any. So then someone at MIT created SnakeMake which is written in Python, as one option. They started doing this, and at the same time, people created Nextflow in Java. Then, in my opinion, I've used both, but Nextflow, yeah, is just a more robust language, and then workflow start to use. And even Stat Central, there was one of the professors that asked us to run a SnakeMake pipeline actually, and that was a nightmare. We're still we managed to do it, but we're still trying to figure it out. A few issues for specific modules. It just makes it harder to troubleshoot a few of the modules. And in fact, we started translating that to Nextflow for our own service to make sure that that would work better next time that we have a professor that wants that pipeline. So yeah, that's actually something that we're doing ourselves. So we're translating that SnakeMake pipeline into Nextflow. Yeah, because we think it's just a stronger workflow language to use. Yeah. No more questions. That's it. Thanks very much for coming, and have a good afternoon, everyone.